There we go. Yes. Okay. Good evening, everyone. And thank you for your patience. Welcome to what is this special session sponsored by the Northern Fund Committee. And what this initiative is a number of years ago, the Northern Fund Committee met and we decided, after spending tens of millions of dollars to advance work on Pacific salmon research, monitoring, and assessment in the northern portions of the Pacific Salmon Treaty Geographic Area, we felt that it was probably important to start showcasing some of the excellent projects that we've been funding, and importantly, some of the excellent work that's been advanced through these important investments. I should introduce myself, Steve Gotcha. I currently serve as the Canadian chair of the Northern Fund Committee, and this evening I have with me
I'm Bill Auger, and I'm not the chair, but I get sucked into it all the time. But these are good to see, because even though I'm a panel fund member, there's lots of times I have no idea what they do with the money. So it's nice to get a slideshow.
Fantastic. Thank you, Bill. So to give us a little bit of context, I think most of you are aware that the Northern and Southern Endowment Funds were established in 1999 on the renewal of the Pacific Salmon Treaty at that time. Their ultimate purpose is to invest in initiatives and programs that build improved information for fisheries management, to invest in rehabilitation of both marine and freshwater salmon habitats, to support programs and projects to enhance wild salmon production, and ultimately to advance our broader understanding and knowledge of Pacific salmon and Pacific salmon ecosystems under the Pacific Salmon Treaty umbrella. And since inception, I'm always impressed to see the statistics that the Northern and Southern funds have enabled over 1,700 projects to proceed and expended over $135 million US in this important work, which I think under today's exchange rate is about 1 billion Canadian, if the math is correct. So we do also take the opportunity to post reports on results from all of the project investments on the Pacific Salmon Commission Endowment Fund webpage. And this evening, it's our great pleasure to showcase an exceptional individual and an exceptional initiative that's been undertaken in Northwestern British Columbia. So we'll be hearing from Mr. Ryan Whitmore from Fisheries and Oceans Canada, who has received, and we're keeping very close track of this, over $300,000 Canadian from the endowment funds since 2019. And tonight we're going to find out what he's done with $300,000. And specifically, he will be showcasing his work on using terminal genetic stock identification and upriver mark recapture to estimate, we'll call it Coho salmon in the Skeena River system. So welcome, Ryan, and thank you for your willingness to present the results of your work.
Yeah, awesome. Yep. Thank you, Steve. Thank you very much. Yes. Yeah, okay. Yeah, well, again, yeah, thanks for the Northern Fund for inviting me up to give you folks a little talk about some of the work we've been doing. I know the folks in the genetic realm have seen this a few times, well, versions of it, and then also NBTC. So excuse me, you're getting a lot longer version of what you usually get tonight. So you're welcome. Yeah, so basically, I'm not going to read the title. Steve did a great job. You know, we're basically just using GSI and expanding it with a mark recapture estimate to get, system-wide escapement estimates for the Skeena River watershed, but then, pushing it further to estimate to the CU and even genetic unit level. And this is work that I've been very privileged to have talked David Hankin to step in and work on this. And when he found out I was giving this presentation, he actually wanted to come, but fortunately for him, he's retired and went to New Zealand last week. But he did want to say that, he used to sit on the CSC years ago and wanted to say, wanted me to mention that he had a great time participating in the Pacific Salmon Commission and really enjoyed his time working with Larry Rudder. Okay. Oh, I was going to go on the next slide. Yes, yeah. So before I really jump into this, here's a map of northern British Columbia. So does everybody here know where that is? Okay. Yes. Nice. Okay. So this is the PFMA areas 1 through 10, which covers northern British Columbia, right? And our DFO office is in Prince Rupert, right up there. And for North Coast stock assessment, for which I work, there are 3 species biologists who are the lead representatives for the government on these files, on their specific files in these 10 PFMAs. So for me, my file that I'm currently in charge of is the Coho salmon file. We have Chelsea May, who is here, who is our representative for the Chinook file, and then Charmaine Carr Harris, who is the rep for the sockeye file. So, going back for Coho or just any of us, you look at this map and you're like, okay, well, that's a lot. And someone dumps it on your lap and says, well, figure it out, Ryan, how many fish are coming back? And you look into it and you're like, okay, well, there's 17 CUs, roughly 900 spawning areas or counting sites according to Canada's open source database. And you ask yourself, where do you focus? Just the basics, right? And for me, fortunately, the Coho file, hasn't really been pushed that hard recently, and it was easy. Focus on the largest watershed in your area. Just step 1. And for us, it's the Skeena River, which a lot of you are familiar with. So I won't explain too much about that, but it is, the second largest watershed within the province, roughly 54,000 square kilometers, has over 200 spawning locations or counting sites of Coho within the watershed. And it's mostly helicopter access. Now, I know people are like, oh, it's not that remote compared to our area, blah, blah, but it is a huge watershed. We have a highway runs about, only about 50% of it. And if you're trying to get up to the headwaters and count or let's say you do an AUC up there with helicopter counts, that's a $10,000 ride every time you go up there. And that's after you spend money slinging fuel up there before the season as well. So if you want to count 2 or 3 populations up there 3 times and get, I'm not a huge fan of Coho aerial counts, but if you want to do it, you're going to spend a lot of money. And that's just a couple small tributaries within this vast watershed, right? So you need to ask yourself, how would you feasibly estimate escapement to a watershed this size? Because you can't go count all that stuff. And they all act a lot different. But before I dig into how we do it, it's more of the why, right? So this is the Skeena River watershed, and until we started working on this project, there was never a system-wide escapement estimate for it. And we formed the first one a few years ago. And why would you even want to do that? Well, it'd be nice to have escapement goals, TRPs, LRPs, BEGs, whatever you want to call them. The first step is figuring out how many fish do we have, or at least estimating, or at least trying to figure out that question. So how would you do this? Well, it's real easy. Piggyback on someone else's work, right? So they've been estimating the aggregate Chinook abundance at the Skeena since 2010, and it was most recently described in a DFO tech report, Winter et al. 2024. The only known publication, or the one I always refer to, is Hamazaki Dekovic 2014, Yukon River Chinook, and then amongst others. And there's this method that I'm going to walk you through here has been used, is being used to the north of us, to the south of us. Folks like to always want to expand using genetics to see what's going on, and a lot of folks, frankly, in my opinion, are abusing it, including me, which I'll show you. But it's being used quite often. And overall, it's just a modification to the simple 2-occasion Lincoln-Peterson model there. So like just another example of someone like, mutilating that model that a lot of us use when we're estimating escapement. So I wanted to show you just the comparison of the traditional mark recapture versus this modification, right? So I have 2 maps here, the Skeena River, and just the arrows for the river entry. So in your simple 2-occasion model, you typically apply marks first. Well, in the traditional method, you mark fish as they enter the river, what have you. In the modified version, the marked fish are already way upstream. And then your second sampling occasion, on your traditional method, you're usually recapturing them or resighting them way upriver from where you marked them. Well, in this method, they're actually downstream the second occasion, right? So it's kind of confusing. It's flipped. A lot of people who have used this in the past like to call this the inside out mark recapture, the upside down mark recapture. I've heard all sorts of names. And when I first brought Dave into this, he got all flustered and was like, "No, that's not true. You can't do that. You can't call it that. It's a simple play on expanding genetic stock identification, because with this method, the modification that we use here, or other folks have used, is that typically the number of marks that you have applied and recaptured are estimates themselves. They're not actually known. So for a project, for this, like this to be executed, there are some major components that need to occur, right? So it seems really simple when you look at that Lincoln-Peterson. It's just like, oh yeah, 2 occasions, but there's some massive projects that I'm going to try to walk you through today, just to kind of tell you, just to give you an idea of what actually occurs. So first, you need to have a mixed stock fishery at or near river entry, okay? And for us, it's the Hyatt test fishery. You need a robust genetic baseline that basically describes the genetic differentiation across all populations within that watershed. Then you need an escapement estimate of a component stock. And for us, this is the Woodset mark recapture program. And then lastly, you need an estimator, right? So I'm going to walk you through each of these components very briefly, because some of these are huge, like the Hyatt test fishery, a lot of you folks know about. I mean, we could have a whole conference about it, right? So, yeah, let's start with this. Okay, so here's a locator map of the Skeena. You can see the Hyatt test fisheries down right near the mouth. And, this is where we do, for this component of the project, this is where we're doing our genetic stock identification. It's a standardized test fishery that's been operating for roughly 70 years. Its primary objective is an in-season sockeye abundance indicator. And for the most part, it's been operating consistently from mid-June all the way through late September. For the operation, so it's a gill net vessel that deploys a net 2 to 3 times daily at high and low water slack tides, only in daylight hours. And this is done in standardized locations relative to tidal flow. The net that they use is a multi-panel gill net with varying mesh sizes that varies from 3.5 to 8.5 inches. They're randomly placed throughout the net, and this net is 200 fathoms by 25 feet deep. And just note that this net is replaced annually. So on an annual basis, the panels are moved. I forget even how they select where the panelings go, but just know that, yeah, it's changed annually. And with this, it's assumed to generate an unbiased relative indice for all salmonids passing through the entire run. So that's the large assumption we make here. For sampling, so all sockeye, chinook, coho, pink, chum, and steelhead that are deceased are harvested. Sampling protocols vary across all the different species. In 2019, prior to 2019, when we first started working on this, coho were a low priority. Remember that it's the sockeye chinook project, and only scales were collected, and not every fish was sampled. It was just kind of hit and miss. And since then, I modified the protocol, every single fish is sampled, and we have paired scales with tissue. And since then, our sample size is roughly 377 fish. And to increase our sample size, I also modified the SOP to collect fish bits, which, you know, the captain of the boat, they're not, you know, my name's not too popular there. So when they bring the net in, if there's parts of fish, you know, that, you know, pinnipeds have been preying on, there's a head or a tail or a perculum or an eyeball even, we have the crew throw them into a bucket. End of the day, they either put them in vials of alcohol or on Wattman sheets for these unidentifiable fish. And when they're genotyped, if they're assigned back as Coho, we can add them to our sample size here. And it's just an attempt to increase it because some years, you know, the sample size is problematic. Okay, so that's the Tyee test fishery. We have our sample size, roughly 377 fish on average for the last 6 years. So now we need the robust genetic baseline. So the SNP panel was developed for Coho salmon. It's described in, you know, Beecham et al. 2020, and it was using archive samples from the late 90s to 2019. And before I go into you know, what we've been doing to the baseline, I need to point out that this was a large collaboration between Fisheries and Oceans Canada, Washington Department of Fish and Wildlife, and Alaska Department of Fish and Wildlife. And as far as I know, it was the first species that had that kind of collaboration, right? I mean, or did like that, like where the SNPs were shared. Well, most of them were shared across bilaterally to build a panel or, let's say, baseline to where, in theory, if fish are encountered in mixed stock fisheries, we should get similar results. But playing with the baseline. So I'm not sure if anybody here has collected genetic samples for baselines or really looked into it, but when I started to look into it, it was troublesome. So, you know, going through our original samples, we found duplicate sampling locations with different names. So you would see the same, you would see two river names, you see them on the dendrogram, because the lab's, you know, not familiar with all of this, like some of us that are in the field. And you'd be like, well, that's the exact same sampling location. That's the same weir, but a different person submitted it a different year, so they called it something different, right? So if you don't look into that stuff, that's how some of your results come back. So we collapsed all of those. Then we resample collections where origin was unknown or unverified. So I think until 2019, most Coho information, you just had the river name, not where it was collected, not any kind of morphometric or meristic information accompanied it. So if it was unknown or unreliable, we're replacing it, or we did. Then resample collections where juvenile genotypes represent the populations, and this is based on Anderson and Waple's 2017 recommendations. And then finally, you know, you're adding populations not currently in the baseline. So here's a map of just all the different locations where we currently have samples representing the Skeena River watershed. So as of 2024, we have 32 populations represented by roughly 3,200 individuals. So we needed to evaluate the performance of the baseline, okay? So this is following Barclay 2019, you know, their methods to evaluate the performance of their panel or of their baseline, right? And this is an example that the lab ran for me where we broke it down in 4 reporting units, which we have the 3 CUs above Tyee, which is the lower Skeena, the middle Skeena, upper Skeena, and then we pull the Bulkley out, and I'll explain that in a little bit. And I'm not going to go into too much detail here. Just know that, you know, according to the methodology of the folks who put this together, their basic guidance was that 90% of the squiggly line needs to be in the diagonal. Everybody got that? So so if you look at that, the squiggly line, no, yeah. So for the mixtures assignments, 90% had to be within 10% of the true mixture, and so only one of those performs at that level. And you can see that a lot of these do not, they kind of fall off here. This one at 50%, this one almost at 25%. And when you look at these colors and you look over at the dendrogram on the left, you can see that the one that's really looks like it's causing a lot of problems is the green one here. And you can see it's kind of bleeding through to all the other CUs. It's not clumped together at all. And so, you know, this would be an area for us to focus on when we're working on it. But the overall picture, or I wanted to share about this, was that the baseline, it's performing decently. There is room for improvement. But, you know, it's okay at this time. But, you know, for someone like me, like I'd like to see, you know, it perform a lot better if we're going to start relying on all these estimates.
Hey, Ryan, what's on the x-axis?
What's on the x-axis right here? Yeah. Oh, okay. So these are mixture proportions.
So as it goes over time, the mixture performance.
No, actually, the proportions are the same on the x and the y axes, right? So you have 50% of the mixture proportions as they come up here. You can see that when you hit 50%, they kind of fall down below the actual mean there. Does that help you a little bit? Because if I get too far into this,
why as it goes to the right, it falls further?
Oh, excuse me.
So it's known and inferred. So known proportions on the X, inferred on the Y.
Oh, got it, okay.
We're testing the
Yes.
baseline against itself.
Got it, okay, got it, okay.
okay. So now we have a baseline. I wouldn't say it's robust at this time, but we have a baseline, 480 SNPs, 32 populations. So let's go into the estimating the component stock. So this is the WITSET mark and capture program. Okay, so we need to have a population of known size here, right? The WITSET market capture program is located, you know, right outside the WITSET village, outside of Smithers. It estimates escapement for Coho, sockeye, and steelhead. It's considered the largest population of Coho within the Skeena River watershed. It's operated by the Office of Wet'suwet'en Fishery Department. And again, it's another use of the simple two occasion Lincoln-Peterson model. So here is an aerial view of the canyon where they conduct the first sampling occasion where they apply the marks. So down here below the canyon, they have a jet boat with a 300-foot seine net that the crews go out, they seine the fish in, tag them, and release them. And then there's also another area in the canyon that they apply marks using dip nets. So the folks will be out there, dip net fish. If they have them, they yell for someone. Someone runs down with a basket or like a cradle. They put the fish in, they run up, they tag them, they release them above the canyon. So just a quick, very brief overview. This is the first occasion where they're marking fish. And then they're recaptured roughly 11 kilometers upstream on the Bulkley from the WITSET Canyon on Toboggan Creek. So this is a concrete weir structure operated by Fisheries and Oceans Canada. And it's considered a census of the entire returning population to this tributary. So all fish are examined, recaptures, recorded, what have you. Then you have your simple Lincoln-Peterson model. This program has been operating for roughly 25 years. And then when we decided to kind of dig in and try to get this escapement estimate for the aggregate, I thought, well, no one's really actually looked at the data for 25 years, but they've been collecting it and summarizing it. But, let's dig in and find out what's going on. So we coaxed another smart person in, so Carl Schwartz, to come in and do a full review for us on that. He has completed his part, and now we're trying to internally finalize the review by the summer of 2025. And it's a full operational and data review for the everything that's available. Okay, so the estimate is estimated. So let's talk about the estimator. Okay, so yes, fish math in hat. Okay, so how do you estimate total abundance? Okay, so you can determine the proportion of your target population at your mixed stock fishery, right? So that was the Tahi test fishery. You genotype those individuals using Rubius. You estimate escapement of your upriver population, and then you plug them into your system-wide abundance estimate. So since the Bulkley escapement estimate and the proportions at Tyee are independent variables, you can use the delta method of approximation. Now, sorry to put this up here. I do apologize. But when Dave said he couldn't make it to come hang out for this presentation, he said, Ryan, I'm going to send you some equations and slides that'll get people really excited because I know how to get a crowd going. He really said that. So, but just to say, it only gets better.
It's an algorithm.
Yeah, I know it's good. Okay, so here's an example of 2024. It's just a map showing you where the different programs occur. So you have your estimated escapement of the Bulkley River, 28,000. You have your proportion of the Bulkley River at the Tyee test fishery catch, which is only roughly 11%. So if any of you actually dabble in this method already, you know that that's not that great, and I'll kind of touch on that in a little bit. And then you have your escapement estimate for the system. So 259,000 fish in 2024. Just seems that simple, right? So this is our 6th year of creating estimates. I only have 6 data points, so I still put them in a table. I can't do a graph yet, so I'm gonna wait a couple more years. But yeah, so you have the year on the left here. This is your escapement estimate, and then the CV, and you can see that the CV is all over the place. You have 40% here, 8% here. This year was 19%. And you just need to note that when you're estimating the variance, it's dependent on the variance of 1, your genetic assignments, and 2, your mark recapture estimate. So these are the required assumptions to make this work. So your population of known size needs to be accurately estimated or genetically different from all the populations within the watershed. Your mixed stock fishery needs to be a random subsample of the entire return. And then lastly, your terminal harvest needs to be negligible, or stock-specific harvest is equal across time, across all subpopulations. So if we do that, can we estimate stock component abundance? And the answer is yes. So since we have the total system escapement, like I just talked about, if we multiply that against the stock of interest, you can estimate that stock, right? But first you have to determine the level of resolution that you either you want or that you can achieve. And is that at the CU level? Is it at the genetic unit level? That's what you have to decide on. And second, you need to estimate the variance and covariance around these, right? And I'll dig into that just a little bit there in a minute. Okay, so this is another one, but so using the delta method, you can estimate the variance of unit-specific escapements. But the issue is that Rubius does not, the GSI program does not estimate the covariance of the stock unit groupings. Yeah, it doesn't estimate it. So we had to figure out a way how to do that, right? So I'll walk you through this fast because this is actually a really wordy slide. So since Rubius does not estimate the covariance, we had to come up with the bootstrap, blow up method, right? Where since you have the population of, you have your aggregate estimate here. Where am I at here? Oops. Yeah, so you're going to create a population equal to the size that we just estimated by blowing it up to the factor of your escapement estimate divided by your sample size at the Tyee test fishery. You're going to select a random sample of small n here, which again is your sample size at the Tyee test fishery, and use Rubius to estimate stock specific proportions. Now you're going to do this a large number of times, and we're finding that 10,000 iterations was sufficient. And I wanted to thank Eric Rondo for doing this for us, because On an annual basis, this is roughly 4 to 6 days of computation time. And for each scenario, because we run a whole bunch of scenarios, it takes roughly, what, 200 to 300 core hours per scenario, right? So they run and we stopped at 10,000 because it appeared to work fine, but we were trying 25 and it kept crashing his computer or his supercomputer. Okay, so then it pumps out a variance covariance matrix. So then you have on the main diagonal your estimated variance, and then you have the off diagonal elements are your pairwise covariance. So very wordy thing there. Okay, so we have this is an example that Dave shared of the 2021 the CU examples. So you have here, we have the CUs, and don't let that confuse you because we have the 3 CUs and bulkily separated. We have their estimated proportions at the tide test fishery as Rubius did. And then we have the Rubius standard errors versus the bootstrap method. And as you can see here, they agree quite well. And Dave was really excited about this. He was really excited. He goes, you know, and I said, and he goes, Ryan, no one will appreciate this at all except for Eric Anderson or the one oddball statistician. But he was really excited that this bootstrap method was able to estimate the variance just as well as Rubius using yes. Are you the statistician? But so this allows us to infer that the covariances are also must also work as well. Okay, so here's a map of the Skeena broken down by its CUs. So you have the lower Skeena, middle Skeena, and then the Bulkley is shaded in blue here because it's within the middle Skeena CU. Then your upper Skeena the conservation unit. It says conservation unit as defined in Holby Serena 2007. Yeah, and so we're able to estimate escapement to CU. So the thing to note here is that the middle Skeena CU is made up of 2 estimates, right? So we have the Bulkley marker capture here, which is 28,000. You combine that with the estimate from the genetic method, you know, to get your full middle Skeena CU escapement estimate. So these CVs aren't the greatest, but you have to keep in mind that, you know, we're estimating escapement to an area, let's say like up here, you know, without even stepping foot, you know, in the region. And it's pretty exciting that you can kind of figure that kind of stuff out. But again, the CUs are problematic. The CVs are problematic. Okay, so let's dive in this a little further. Can we estimate to genetic units? Okay, so how far can we push the baseline to break it up into so many genetic units that we're still confident that we can assign to them properly, right? And for us, you know, individual assignment accuracy had to be greater than 85% for it to be its own genetic unit. So an example here is the Babine watershed. So that pulled off on itself, or like the Bulkley Rivers, this pink one right here. But for large genetic units, we had to collapse them into geographically proximate populations to achieve the accuracy for us to separate them properly. An example of that would be like, you know, upper Skeena, where these 3 watersheds in the baseline had to keep collapsing until we met that level of accuracy. So then we were able to break it down in 14 different genetic groupings. And so you can see here, this is the name of the genetic grouping. This is your escapement estimate, and this is your CV. But I want to highlight this. This is for discussion only. If you were to take this information and share it or use it for management at this time, you'd be committing what we call crimes against rep units. Okay, so because it's all over the place. So you can see here at the Babine watershed, we had an estimate of roughly 5,500 with a CV of 19%. Not so bad. And then you come down, you know, to the Motazi River, we have 1,400 fish with a CV of 102%. Like not okay, right? So again, if you were to report this to management, not good. Don't use it. And there could be many factors around of why this is occurring. Is your sample size large enough to detect these rare stocks? Because again, we had an escapement estimate of 250,000. You had a population of 1,400 here. Was it large enough? Probably not. And also, there's, when you're looking at mixed stock analyses, fish aren't assigned, to the population 100%. There's fractions of percentages for each of those individuals. And we like to call them low but non-zero probabilities. And each of those low probabilities have associated error. And when you add those up and you blow it up like this, you get crazy estimates, so, and this is, again, this is an analogy of like a mixed stock. This is a mixed stock fishery, right? So you apply this across. So if you're not actually detecting individuals and you look at the variance there, it's scary. Yeah, anyways, but this shows you what we can do, but I'm not saying we should do it. So what are our future considerations here? The one thing I've kind of focused on here is this we need to reduce the variance in some of these situations. And how would you do that? So, one step is to improve the genetic assignment. So number one, continue to refine the genetic baseline as we described earlier. But also what we're proposing to do is create a Skeena watershed specific SNP panel. So the thought here is you create a very specific SNP panel for the Skeena that should reduce the uncertainty around the assignments to a negligible level. And then when we ran a few simulations for a few years saying like, oh, if you remove the uncertainty in your assignments, how would that change your variance? And it would change it up to 5, it would reduce the CV by up to 5%. And I have the asterisks there because there's a lot more to it than that, right? So we have a sample size issue. You always got to be careful about, how far you want to break into it, depending on your sample sizes. But, if we're able to achieve this and complete that, we can finally finish the genetic grouping delineations. And again, it's dependent on the genetic differentiation amongst your populations, but ultimately it's your sample size. We're back to sample size again, because I could break it down by, let's say that we're able to break it down by every single population. Well, if I don't have a very large sample size of Tahi test fishery, I'll get those really weird estimates like I showed you at the Motazi River. So there's a lot to think about here when you're doing genetics and you're playing with this kind of stuff. And I always, recommend, asking the geneticist to review your information before sharing it. Okay, so then, and also we can improve the WITSET mark recapture estimates, because again, that's the other variable of uncertainty. And so our program review should be done in the spring, summer of this year. And we're also, trying to increase the capacity for that group to have more people out there, because it needs improvements all around. And then lastly, what if you increase the number of populations of known size, right? So I didn't want to go into this because this has already been, really long. So so my thought was, like, this year we had, the Bulkley River represented only 11%. What if I took another population of known size? So like, let's leverage another program like the Babine Fence. There's a couple other small market capture programs within the Skeena. So if you take those estimates and you combine them with the Bulkley, what does that do to your estimate? And the Babine's a good example because we have a census of all Coho returning there. So we're actually increasing the population size, and we don't add any variance around that because it is a census. And just for this year alone, when I combine the Babine for the aggregate escapement estimate, it dropped the CV from 19 to 16%, right? So, how far can we do that? Would this lead us to start other fence programs to back up the Bulkley River market capture program if we need to? So it's something else that we are considering and looking at as well. And yeah, that's it. For acknowledgements here, I've talked, thank the Northern Fund for dealing with me and giving me $356,495.33. And the office of the Wet'suwet'en, for their crew out there doing that, you saw those folks, they're tethered to rocks with dip nets to catch these coho and tag them for us. And then, all of our staff for working on that stuff. Yeah. And yep, that's it.
Ryan, well, thank you very much for the presentation, and just want to acknowledge your solution-oriented and creative approach to this, what I would describe as cutting-edge research and monitoring in the north coast of British Columbia. Happy to entertain questions from our audience, starting with Commissioner Vincent Lang.
Hey, how you doing, Ryan?
Good.
That's a good presentation. So best I could tell, you have a good estimate of the fish coming into the overall system.
Mhm.
Then you have one stream and you estimate, the genetics of that stream, then you go back and you estimate that proportion to kind of give an estimate of what's going through. So that seems to be pretty reliant on the fact that that one system is a constant proportion over time of the rest of the system. And how accurate is that assumption? And then number two, as you start splitting that out amongst a bunch of different subsystems, that seems to also indicate that you need to have constant proportions of all those different systems doing it. And my experience with Coho is that there's a lot of variability within a major rivers like the Susittan River in Alaska between all those different stocks each year, which some produce really high and some produce really low. There's no constant proportion. That's 15% a year.
Yeah, so I wouldn't say you need a constant proportion. Just know that, as your proportion goes down, your variance goes up, right? So if some of those years, like for example, a lot of clicking here. So some of these years,
I thought the answer would be in that form.
so right here, no, it's yeah, nice one. Yeah, so for like, where was I going at with this? So like, yeah, for like most of these years, let's say in 2023, the bulkly represented 26%, right? This year it represented 11%. So your uncertainty increases as the proportion goes down, right?
Okay.
And with the other populations, like as you expand it and blow it up and get some of those wild estimates, it does get crazier as that proportion goes down, like as I was talking about there.
Thanks.
Yeah. Yep. Thank you.
Good work.
Thanks.
Do you trust Hyatt or to give you that proportion? Because, one of the problems with smaller, one of the problems with smaller species or stocks, depending what you're doing, is that if you had a large sockeye run or a large pink run, the net becomes saturated. And so coho and steelhead, for that matter, are phenomenal avoiders, right? So I'm just wondering if you have noticed a difference in years of high sockeye. Like if you look at 2023, that was a pretty good sockeye year, right? So have you noticed, Ryan, a difference when that net is getting plugged with sockeye or not?
I have not, and I haven't looked in that too much. But yes, I fully understand. We've chatted with that a lot about, nets.
Because that's what Dave Peacock and that crew in my youth said, that it was difficult to use a tie to as an assessment tool for smaller populations, because when sockeye, when the net becomes full, or even if the net's not, if it's a bigger fish, then you got a seal problem. But anyway, I just want to know if you noticed that, because I just looked at that 2023 number. It looks pretty solid at 0.08, but that's when the net was, we had a pretty big run of sockeye that year.
And we also had the record catch at Tyee for Coho that year,
Right.
actually, as well. It was double the average. Last year was a stellar year for Coho. Well, there's quite a few factors there. So, like I said, I haven't really looked into it, but the run timing of Coho and sockeye do not exactly overlap as well, right? So at the peak of the sockeye is not the peak of the Coho run, right? So you have that to think about. Yeah, and that'd be a tough one to tease apart. You know, the one thing we've done to test this to see if the estimates make any sense is you do a back calculation. And I didn't have this in a pocket side. I thought about putting it up is when you use different proportions to estimate different populations. So I could use the proportion of the Babine with the Babine census count to estimate the Bulkley River mark recapture and see how close it was, right? Or vice versa. And we played with that. Some years it works, some years it doesn't. But that's an assumption, you know, moving forward in this we have to make is that, like I said, underline, it's assumed that we're getting an unbiased subsample of the entire population. Yeah.
Further questions, comments? Front of the room?
I'll just shout. So for the 480 Smith baseline, were any Skeena River populations used for developing that?
I don't know that answer. That's an easy one for me. Eric, save me.
The Coho baseline was built in 2 stages. First stage, probably not. The second stage was developed in Regency for the project. And that included coastwide populations, including Skeena. And I think actually many of those SNPs were used to boost the capabilities on the North Coast range because most of the original panel was based on development on the South Coast. So the answer is yes, but we do think that further focused effort on the Skeena specifically is still required in this case to boost those numbers.
I had a quick question about the program review on the market capture that you're basing a lot of this on. Is just, could you share like preliminary results of that program review, what Dr. Schwartz found, and discuss briefly about what maybe the results of that program review would be like implications for your estimate?
Ooh, that's a long one. Yeah, the review, well, you know, there are multiple parts there. So the big review is that, you know, and I didn't get into it, is that, you know, at the WITSET market capture program, we do individual identification tags for every single fish. So we have IEHs for those. We also have weekly bins and all that. And we were trying to utilize maybe like a more robust model besides the simple Lincoln Peterson pulled model with the Chapman modification, right? So we were trying to take all that information to see if you could find something more reliable or with like a more, what I would consider a more realistic estimate of uncertainty, because we know what the Peterson model does. So that was like the main focus of the Carl Schwartz review. And what he found out was that it's a real mess. And that's why it's taken 3 years for us to figure out what's going on. So the results right now were for us to be more involved in the programs. So since his review, you know, we're a lot more involved in the Toboggan Creek fence. That used to be a contractor that did that for us. So we're kind of stepping in and running it because we thought there were issues in data recording. So everything from trying to start trying to develop a new model to just basic operations, right? So right now we ran 4 different models and we're trying to decide which ones to use. Because, you know, we're not going to use the one with just the lowest variance because, right, that's cheating, but it's, you know, which one makes sense and which one's the most reliable and repeatable over time. That answer that decently? Okay, cool.
I have a question going back to the SNP.
Yes.
We're working in the transboundary, particularly on the Taku, but also Skeena on developing the Coho baseline as well. And so I'm really interested in the panel being representative and wondering your interest in maybe looking at a SNP panel just for the Skeena. Is that the level of approach we need to look at for individual rivers, or can we leverage that effort with other efforts that we'll be perhaps needing on the Taku, where we could improve the coastwide baseline?
What do you think there, Eric? I mean, so like, well, like, so I think it matters what your objective is. It's like, what do you want to see? I mean, are assignments to the CU level in the TACU, is that your objective? That will yield a different answer to where like we really want to really look as precise as we can to break up as many units as we can with confidence in those assignments. So it just matters what you're trying to do. But the coast-wide baseline, I mean, that's something that also, you know, we've been supported, a lot of us here by the Northern Fund to keep working on that as well, right? So I don't know, Eric, that's my answer.
Yeah, I would say it probably depends on the objectives. Ryan has a really clear objective, what he's trying to recover. And as we kind of demonstrate there, there's room for improvement. And we do see the genetic differentiation within the system based on the existing data to suggest that perhaps tweaking the SNPs that are included or the markers that are included should yield gains in the assignment power for those component groupings that he's targeting. So as we expand out into TACU and establish that baseline using the existing marker panel, I think the next question will be, what differentiation is there, and are we able to assign to the units that are required for that? Now, there is the counterpoint where at some point there will not be sufficient genetic differentiation just based on the system. We see that elsewhere. We attempted a similar thing in the Fraser River for Coho, Sockeye, and Chinook. And for Sockeye, it was beneficial. And for Sockeye, for Chinook, it was beneficial. For Sockeye, we'd hit the limit of what was possible, we think. So it's more markers can be good, but more markers isn't always a silver bullet. But circling back, I think the main thing will be, what is the objective? What do we need to separate out? And if we can't do that with the existing panel, then perhaps a similar effort could be required.
So you're suggesting a coast-wide whole genome panel, right, Kyle? Next year's collaborative proposal?
Any final questions for Ryan?
Is there a Northern Fund sign posted outside of one of your workstations?
That's not even no, not yet.
Great segue to express our appreciation to Ryan for the tremendous work that you're doing. And it's an honor for the Northern Endowment Fund to have a small part in our contributions toward that. I know we started off this evening with the question of, where has the 300,000 gone? And the answer came to me in Ryan's presentation, and that was in the form of the 10,000 simulations. And I know Bill Otter would be the first to say, where can you get a good simulation for $35? And you think Ryan Whitmore. With that, thank you very much, Ryan. Thank you to our audience for attending this evening's session. And if you do have any further follow-up questions for Ryan, feel free to reach out to him directly. If you have an interest in understanding how you can apply for funding from either the Northern or the Southern Endowment Fund committees, please visit the Pacific Salmon Commission's website. We do our call out typically around the May period of each year, and look forward to hopefully seeing continuation of great work, such as what Ryan showcased for us tonight, and other project proponents in future investments of the Northern and Southern Endowment Funds. With that, thank you very much for your attendance and participation this evening.
Good enough, went smooth.